This is video number 6 in our series about myopia. It is an overview presenting highlights from our 5 in-depth subject videos, so I'm calling this one The World of Myopia. Myopia, or nearsightedness, is well known as a cause of blurry distance vision. Compare the sharp image on the left to the blurred image on the right. However, it has a less well-known side, risks it poses to eye health. For example, it is associated with retinal detachment and a form of macular degeneration. The frequency of myopia has been increasing around the world, in some places reaching over 3/4 of the population, and that rise is expected to continue. With the risk of increasing vision loss, it is attracting a lot of attention. In this summary video, we are going to take highlights from the series videos to create a general overview. The series videos have a lot more information. They include, number 1, what myopia is and how widespread it is. Number 2, what risks it poses to eye health. Number 3, research to understand how the eye controls its own growth. Number 4, some eyes diverge from stability and grow into myopia. And number 5, development of treatment options and their effectiveness. That is our series. And these are the subjects of this summary video, with times so you can move around if you wish. Before we go further, a word about our approach to this subject. Even though myopia has been under study for several decades, in some ways it is still a developing field. Figuring out the cause or causes remains a work in progress. Likewise, when to treat and how to treat are also works in progress. Because this is an evolving field, I have chosen a study-based approach, presenting mostly landmark studies, comparing and contrasting results, including disagreement between authorities. Hopefully, you will find this as interesting as I do, following researchers as they work to figure out the puzzle of myopia. What is myopia? We will start with a brief review. In the sense that your eye works like a camera, if you had ideal vision, light rays from a distant object would come to a focus and make a sharp image on the retina in the back of the eye. This is called emmetropia. In people with myopia, the eyeball is longer than average. One result of which is that light comes to a focus in front of the retina, making a blurred image on the retina. As we showed before, an example of the sharp image of the emmetropic ideal versus myopic blur. Sharp focus can be restored using a corrective lens, specifically a diverging lens to push the image back to the retina. The other result of the longer eyeball is that the layers of the eye are stretched thinner, causing problems in each of them. These are the main complications of myopia. The impact of myopia, then, is vision loss from either refractive error or pathology. With a vision chart for perspective, on the right are criteria for vision impairment and blindness from the US and WHO. They differ slightly. In 2013, the leading cause of blindness worldwide was cataract, and not far behind, uncorrected refractive error, which means myopes without distance glasses and presbyopes without reading glasses. Regarding moderate to severe vision impairment, uncorrected refractive error accounted for a little more than half. Such reduced vision impact people's daily lives, like school performance and job productivity. How many people have myopia? Numerous studies show the percentage of people with myopia is increasing around the world. Here, comparing percentages for the year 2000, about 25% worldwide, versus the projection for 2050 at 50% worldwide. For example, in the US, the Vitale study compares National Health Survey data from the early 1970s to the turn of the century. The prevalence of myopia at the beginning of the 70s is about 25% across age groups. 30 years later, myopia has increased in all age groups. For example, an increase of over 18% in the 25 to 34 age group. The dashed line at the top marks 50% of the population, and that was 20 years ago. Compare that to the increase in the East Asian countries, where the incidence is now around 80%. In 2015, Nature carried this news piece headlining an epidemic of myopia. I include it here because it is cited just about everywhere, a sign that this is getting worldwide attention. Before we go into myopia complications, we need to talk more about refraction. The ideal of sharp vision is emmetropia. To the extent eye growth gets off track, the eye can become near or farsighted. The amount the eye is out of focus is called the refractive error. Usually hyperopia resulting from a too short eye, and myopia from a too long eye. The power and sign of the lens needed to bring the image into focus is called the refractive correction. Correction is measured in diopters, seen on the number line. Hyperopia correction is in plus numbers, here to the left of 0. Myopia correction is in minus numbers, to the right of 0. Regarding myopia, some studies specify that myopia formally starts at a spherical equivalent of minus 50, while others choose minus 75. Most studies categorize minus 50 to minus 3 as mild myopia, minus 3 to minus 6 as moderate myopia, and greater than minus 6 as high myopia. WHO puts high myopia at minus 5. These divisions are based both on reduced vision and complication rates. Note the color coding for low, medium, and high, as it will continue through the video. It is a key concept that the risk of complications increases with degree of myopia. In order to understand what goes wrong, you will need some understanding of the structure of the eye. Here I have constructed a conceptual diagram representing the normal eye. It exaggerates the thickness of each of the layers, showing them as even from front to back. The innermost layer is the retina, a layer of nerve tissue that acts like film in a camera. Under that is the choroid, a layer of blood vessels that supplies the outer retina with oxygen and nutrients it needs to function. The outside is the sclera, a tough, fibrous layer that gives the eyeball its structure. The effect of myopia is thinning of each of the different layers of the eye, but not necessarily in the same regions. Myopia-caused thinning is thought to cause these complications. Myopia issues all begin with the sclera. The eye of a child destined to be a myope starts out normal, but as it grows, the sclera thins, particularly at the back, allowing it to stretch or elongate. The first result of the elongated eyeball is refractive myopia. Second, the thinned, weakened sclera is also vulnerable to local bulging, called a staphyloma. This view shows the left half of the eye as normal size and wall thickness, and the right half showing thinning and elongation, causing the refractive error. On the right half eyeball, there is an area of additional localized thinning and bulging, the staphyloma. Like the sclera, the choroid becomes thinner toward the back of the eye. This is associated with atrophy, part of myopic macular degeneration. This is a normal macula and a myopic macula with central atrophy, resulting in loss of central vision. The retina, on the other hand, has normal thickness in the back of the eye, but is thin at the equator. This peripheral thinning increases the chance of retinal tear and detachment, which looks something like this. Lastly, the optic nerve is a bundle of nerve fibers that carries information from the retina to the brain. If the pressure within the eye becomes elevated, that can damage the optic nerve, causing loss of visual field. That's what glaucoma is. Myopia causes changes in the structure of the optic nerve that may predispose to damage at a lower pressure level and or it might be a unique form of optic neuropathy. These then are the main complications of myopia. The risk for developing each of the complications is related to the degree of myopia. A recent review by Harmon shows us how much. These charts are based on odds ratio, where non-myopes have an odds ratio of 1. The association between myopia and myopic macular degeneration is very strong, extending off the chart in high myopia. Retinal detachment rates increase significantly with increasing myopia. Both of these show a pretty big jump when myopia passes -3 diopters, when it moves from mild to moderate. Regarding glaucoma, low myopia shows a modest increase in risk. Here, moderate and high myopia are combined with an almost 3 times risk. What is the chance these complications result in vision impairment? This is the odds ratio of having vision impairment with different levels of myopia. Risk of vision loss is low with low myopia, but increases steadily, roughly doubling with each step up in myopia, until it reaches an extreme at -15 diopters. The maculopathy alone is projected to be an increasing cause of vision loss and blindness in the next few decades. Now, relating myopia levels and risk, back to our world map. The figures from Holden showed us in the year 2000 about 23% of the world was myopic, and just short of 3% was highly myopic, the high-risk group. By the year 2050, almost half the world will be myopic, and almost 10% will be highly myopic, with all the risks that carries. To address the increase in myopia, there has been a lot of investigation into how the eye grows, more specifically, how it grows into myopia. This is the growth curve for average eyeball length from post-conception up to 3 years of age. Birth is at 40 weeks. In the newborn, the average eye is a little over 2 diopters hyperopic, with a moderate range of refractive error. By 9 months, the gold line, the average refractive error is less and the range is narrowed. Something is guiding eye growth, a remarkable process called emmetropization. This is a comparison between newborn and adult eyes, roughly to scale. The cornea and lens follow a pre-programmed path of growth. The mechanism for getting the image to land on the retina as the eye grows depends on fine-tuning the length of the eye. To see how that works, researchers have measured growth in the different parts of the eye. Here is what eye growth of the component parts looks like in emmetropes. In this graph, myopic refraction is downward. The power of the cornea and lens gradually decrease, while axial length increases, in this case synchronized so that refraction remains stable near 0. The key point here is that the eye is guiding its own growth toward emmetropia, an amazing bit of physiology. For comparison, this is emmetropia versus myopia. In myopes, the cornea and lens changes tend to offset one another. The big difference is the increase in axial length, ascending off the top of the chart, while refraction descends off the bottom of the chart. Axial length increase being the primary cause of diverging myopic refraction. Another key point. Research has shown that even though you perceive sharpness in the central retina, rather it is the peripheral retina that is in control of emmetropization. Remarkably, the retina is able to detect whether the image is in focus, and if it is not, which direction is out of focus. So the emmetropization correction process works like this. If the peripheral image is in hyperopic defocus, then growth accelerates to catch up to the image. If the image is in myopic defocus, then eyeball growth slows down to allow the image to catch up. That process involves a signal cascade starting with the retina, signaling to the RPE choroid, which in turn signals to the sclera to remodel. Remodeling means change in the synthesis of collagen, which can slow down or speed up elongation. A minute ago, we saw how well that process works in the newborn eye as the amount of refraction decreases and the range narrows. Another way to look at how well this mechanism works is to ask, what happens if the mechanism isn't present? As a comparison reference, in this survey of 12,000 people, the average refraction was about plus a half diopter with a narrow spread of values. Compare that to people who had some kind of pathology that degraded the retinal image, like cataract or retinal disease. That is represented by the orange line. Without a clear image, feedback control is lost and the eye takes on a wide range of myopia, up to minus 14 diopters in this survey. That normals have such a narrow distribution shows you what an effective mechanism the eye has for regulating its growth. In terms of percentages, at age 3, emmetropization is done very well, with 80% of eyes emmetropic and less than 2% myopic. This holds steady through age 5. But then, around age 5 or 6, something changes and myopia begins to develop. By age 10, emmetropes have decreased and myopes have reached around 50%. Switching to a slightly later age group shows the continued progress of myopia. Presenting the data this way makes it look like myopes all behave as a group, but that is not the case. It turns out onset of myopia happens at different ages. This is significant because earlier onset is associated with a faster rate of change and higher eventual level of myopia. Digging a little deeper, the eventually myopic eye started out as emmetropic and began to progress toward myopia several years before crossing the threshold. Just so that is clear, the myopia group starts to diverge from emmetropia some years before crossing the myopia threshold. A point to consider. If school begins at age 6, the divergence of the early onset myopes begins years ahead of reading and near work. Why is this happening in myopes? Has emmetropization stopped working? Has reduced effect? Or is it overpowered by other factors? That has spurred a search for specific causes of myopia. We know that genetic background accounts for basic susceptibility, but researchers believe the prevalence is increasing faster than can be accounted for by changes in gene frequency. There must be other contributing factors. In our modern world, children spend more time indoors with less exposure to sunlight and less exercise. There is increased near work, as in studying and computer work. And more recently, there is a question of whether there is influence from the way standard glasses correction works regarding central versus peripheral focus. More on that in a minute. Let us work our way through each of these likely causes. We begin with genetic background. In video 1, we took a longer look at inheritance. We will briefly summarize that here. Twin studies showed a high degree of heritability. For a long time, that was the principal evidence for considering myopia to be mostly inherited. In recent decades, there has been a search for specific myopia-related genetic defects. Up to 2010, linkage studies had identified 20 such genes. But keep in mind, these are uncommon individual genetic defects that should be considered separate from the general population of myopia. The identification of myopia-associated genes increased significantly with more recent genome-wide association studies. And genome-wide studies can be combined to give even larger sample sizes. Just such a study was done for myopia, the CREAM study. Screening 2.5 million gene markers, or SNPs, They determined which genes had the highest association with myopia and ranked the strength of association. There were 19 genes with the highest plausibility scores. What do these genes do? The authors created a nice graphic showing where those specific genes are functioning and how widely distributed the various genes are. They are mainly involved in retinal cell physiology and light processing. For example, GNB3 in glutamate signaling, and others in association with the dopamine pathway. However, for complex traits like height or myopia, identified markers or SNPs generally only account for a small fraction of the phenotypic variation expected from twin studies. The 2018 Cream paper, even combined with the 23andMe datasets, only accounted for about 8% of the variation in refraction. That compares to twin studies which found around 80% heritability. That large gap in strength of association has become known as the missing heritability problem, raising a debate about the relative roles of genes, epigenetics, and environment. In a recent review, one authority concludes the genetic contribution appears to be more complex than researchers expected. Which brings us to the other side of the coin, environment. We are going to discuss this in two parts. One will be general population studies. The other will be a look at what factors in the environment could be causing myopia. In 2009, he and co-workers approached the environmental question by surveying children from different cities within China. So it is presumed they have a similar genetic background. Any differences in result would be from urban versus rural environment. The urban cities showed similar high rates of myopia. Compare that to the lower rates of myopia in the rural environment. The authors concluded the urban-rural difference is likely the result of differences in environmental factors, possibly including educational, socioeconomic, and/or nutritional effects. A similar point is made by Morgan, looking at different ethnic groups in Singapore. They assembled studies to compare myopia in different ethnic groups within Singapore versus the same ethnic groups outside of Singapore. The large difference in prevalence adds to the case for environmental influence. The next question, then, is what is it in the environment that causes myopia? The Sydney Myopia Study was an important survey of potential myopia causes. Regarding choice of parents, having more parents with myopia is clearly associated with more myopia in children. Regarding outdoor activity, here the y-axis is increasing hyperopia. This tells you that more outdoor activity is associated with more hyperopia, which is equal to saying more outdoor activity is associated with less myopia. Regarding near work, different levels of near work did not have a significant effect on myopia development. This 3D graph is the most often presented result of the study. It shows the same conclusions we just stated. The authors made a couple of additional comments. First, it was likely that outdoor time was the controlling factor, not sports participation. Indoor sports made no difference in outcome. Second, in addition to the lack of correlation with near activity, they also looked at mid-range, like TV, video games, and computer time, finding no relationship with the final refraction. The Orinda longitudinal study of myopia was done at about the same time in California. They came to the same conclusions about association with myopic parents and hours spent in sports. Reading hours were borderline and dropped out in multifactorial analysis. One interesting analysis involved the effect of outdoor time and sunlight. They found that a child with two myopic parents who spends little time outdoors had a 60% chance of becoming myopic. However, if the child with two myopic parents spent 14 hours a week outside, the risk dropped significantly to the same level as a child who only had one myopic parent. Also note that having 0 parents with myopia does not mean 0 risk for the child. Indeed, in the low exercise group, the risk is about 30%, the same as having one myopic parent. In 2019, prominent researchers Zadnik and Moody published an editorial on their views of myopia causes, near work versus sunlight. Consider this as one side of an expert point-counterpoint argument. They identify outdoor time as important, specifically referencing the 2007 Jones graph. They do not find a convincing case for near work as a major cause of myopia. But puzzling that light exposure does not slow myopia once it is present. They reference this 2017 meta-analysis which found that outdoor time is effective at reducing the onset of myopia, but does not appear to slow development in children who are already myopic. Note, this is before the 2018 Wu result. For the counterpoint view on reading and near work, Morgan gives us a different perspective. In a survey of the world's literature, they found that educational systems that require a large number of hours spent in studying and near work and allow for fewer hours of outside time show a strong relation to increased myopia development. There is one more factor in contention for an environmental cause of myopia development. In the beginning, we discussed that eyeball growth is controlled by emmetropization, based on image focus, not in the fovea, but in the mid-periphery. Specifically, peripheral hyperopic defocus is the leading candidate for triggering increased eye growth. That is an important concept because standard glasses focus the image on the fovea, but may leave the peripheral retina hyperopic. This has become a major avenue of investigation for controlling myopia, motivating trials of multiple versions of glasses and contact lenses that aim to control the peripheral refraction. In an attempt to sort out causative factors, a large multi-center study was instituted, the CLEAR study. It used 5 trial sites with more than 4,500 children involved. In brief, they looked at 13 different likely factors to try and identify if any could be used to predict development of myopia. Of the 10 factors that had a detectable relation to myopia development, 8 survived multivariate models. The author's conclusion was the best predictor of myopia development was baseline refractive error. That single parameter had nearly the equivalent predictive strength of the other 8 variables put together. Following this logic, they suggested a decision to treat could be based on early refractive error. These, then, are the leading contenders for the causes of myopia. Methods used to try and slow development of myopia are aimed at each of these causes, except parents. Treatment methods include various glasses and contact lens options, orthokeratology, therapeutic drops, and increased light exposure. Because this is a summary presentation, we are only going to present one study for each method. The full videos present more studies with, interestingly and not surprisingly, contrasting results. We start with the simplest intervention, glasses options. On the left is a standard single vision lens we are accustomed to correct distance vision. In the rest of the lenses, the orange color represents plus power, an area used for reading in bifocals and a change in peripheral focus in the myopic specialty glasses. The simplest refractive strategy addresses the theory of hyperopic defocus directly by simply undercorrecting the myopia, leaving the eye intentionally myopic. In theory, myopic defocus should result in slower growth. In the Chung study, they undercorrected myopia by 3/4 of a diopter. Note that undercorrecting myopia is the same as making a reading glass. The fully corrected control group shows typical changes in refraction and axial length over the 2 years of the study. The result of undercorrection was a surprise. The undercorrected eyes, the gold line, developed more myopia than the fully corrected eyes. More myopia. Other undercorrection studies have had mixed results. Undercorrection is officially not a recommended treatment strategy. The second treatment strategy starts with the straightforward idea that if reading and near work are the cause of myopia, it is logical to look at the effort of accommodation as the cause. Therefore, if you use reading glasses or bifocals to reduce or eliminate the effort of accommodation, that should reduce development of myopia. The well-known COMET study did this using progressive bifocals. This resulted in a slight reduction in myopia in the first year of 1/4 diopter and no change after that. The authors commented that while this can be measured as a true effect, clinically it is not significant and should not be used. Other bifocal studies have shown mixed results. Option 3. Standard glasses focus the image on the fovea but may leave the peripheral image hyperopic. This has motivated trials of multiple versions of glasses and contact lenses that aim to control the peripheral refraction. About a decade ago, there was a notable trial using this strategy with 3 design variations, differences in the central aperture size and plus power. The study was designed to go for 2 years, but it was stopped after 12 months because of the overall lack of positive effect. Compared to the controls, 2 lens designs showed no improvement and 1 only a small improvement. However, a later trial used glasses that corrected peripheral hyperopia with a different lens design. This trial had more success. Option 4. Increased outdoor time and exposure to sunlight. The He study added 40 minutes of outdoor time on each school day and encouraged more outdoor time in general. In terms of refraction change, there was a small but statistically significant slowing of myopia progress, while for axial length, the change fell just short of statistical significance. Whereas the 2018 Wu study showed a stronger effect. That gave us a look at the methods we might have expected to work based on our understanding of emmetropization. But that is not all. There are 2 more methods, ortho-K contact lenses and atropine drops. Orthokeratology uses a lens made of a rigid material that temporarily molds the cornea with the purpose of changing the glasses prescription. Its effect on myopia development is thought to be through an effect on peripheral refraction. It has had moderate success. The main pharmacologic treatment for myopia is atropine. It has shown significant success in slowing myopia development. An interesting finding is that the low concentration seems to work the best. Video number 5 covers a larger range of studies and has a detailed comparison of the results. For the purpose of this video, in 2016, Wang led a review of the effectiveness of various treatment methods, finding atropine to be the most effective. Again, trials are reviewed in detail in video 5. How then does one make a treatment decision? Who to treat, when, and with what method? There is general agreement that the best predictor of degree of myopia development is what age it starts. The progression of myopia is easy to see in this graph. One approach would be, if you want to keep myopia from ending up over -3, one would consider starting treatment if onset happens before age 8. Another strategy comes from the Adams study using atropine. Their algorithm recommends treating children aged 6 to 12 who showed myopia progression of greater than half a diopter per year. Depending on their response over a 2-year period, one would decide the second step. What is happening in practice? In 2020, Leshno published a survey of pediatric ophthalmologists worldwide. About 57% of them were treating myopia. He asked, what were the criteria to start treatment and what treatments were they using? Results are presented by world region. The green bars show percentage who treat based on age of onset. The numbers at the top are the average age for each region, which range from 5 to 6 years. The gold bars are percent who treat by amount of myopia. By far the most common reason for initiating treatment is rate of myopia progression, the orange bars. The average threshold for treatment was about 1 diopter per year. These are the various methods chosen for treatment. Atropine in red dominates in most of the world except Europe, where optical choices are preferred. Behavioral measures include reduced near work and more time outdoors. Combinations are often employed, as shown here for North America. For perspective on the treatment decision, Bullimore and co-authors offer this risk versus benefit analysis. So far, we have established the risk of vision loss increases with the degree of myopia. Bullimore calculates approximately an extra year of vision impairment for each diopter increase in myopia. Here they get more specific and calculated for each additional diopter of myopia the percent of increased risk for specific eye problems. The complementary observation is that reducing myopia results in less complications. The higher the level of myopia, the greater impact of myopia reduction. Specifically, 1 diopter of myopia reduction can prevent between 0.74 and 1.22 years of visual impairment for myopia from -3 to -8 diopters. Thus, the authors conclude 1 diopter of myopia reduction as meaningful in reducing complications. They have also calculated number needed to treat to reduce vision impairment by 5 years. In terms of treatment methods, these authors estimate that, with current methods, a 1 diopter reduction in myopia would likely take 5 years of treatment. What are the risks of treatment? 2 methods have potential significant side effects. Contact lenses introduce the risk of bacterial infection in the cornea, which can cause scarring and reduced vision. They review the literature and come up with an estimate of low risk. Atropine has a long track record with low risk, as we discussed earlier. The conclusion of these authors is that the benefit of treatment in reducing vision loss outweighs the low risk of vision loss from contact lens use. Given the investment in time and effort required by these methods, it will require significant education for parents, and don't overlook engagement with their myopic children. We conclude our exploration of myopia with a broader perspective from a review by Morgan and an assembly of all-star authors. Here is how these authors analyze the world increase in myopia. There is an epidemic of myopia in Asia with attendant high myopia. High myopia having potential sight-threatening consequences. Because this increase has occurred over a relatively short period of time, it must be acquired, not genetic. They contend the contributing factors appear to be heavy emphasis on near focus and limited time spent outdoors. These risk factors should be modifiable. There are additional measures, for example atropine, that can be used to control myopia. A year later, Morgan and Rose provided a critical review of the genes versus environment debate, including historical background, criticism of study methods and conclusions, and current study results. They discussed the debate about nature versus nurture. First, Environmental factors can have a major impact, while genetic contributions are more limited than was assumed from twin studies. Second, the idea that myopia primarily occurs due to a genetic failure of control over refractive development needs replacement. And finally, it is therefore suggested that school myopia should be regarded as the result of a control mechanism operating in conditions that are so unnatural that myopia occurs as a disease of advanced civilization. As we come to the end of our myopia summary video, we want to make two closing points. We have seen myopia is increasing around the world, in some areas already at epidemic levels. Why care? Partly because the blurred vision needs to be addressed, but also because the pathologic changes pose a risk of vision loss. How much risk? The figures from Holden showed us that by the year 2050, almost half the world will be myopic and almost 10% will be highly myopic, with all the risks that carries. As we said in the beginning, understanding the causes of myopia remains a work in progress, and developing treatments also remains a work in progress, but worth our attention. This video is intended as an overview of myopia, summarizing the concepts which are presented in detail in the 5 subject videos. If you want to know more, in video number 1 we discuss what myopia is and how it is increasing around the world. Video 2 is a discussion of how myopia leads to vision-threatening complications. Video 3 takes us into experimental investigations into how the eye is able to control its own growth, usually. Video 4 explores the divergence of myopes and possible causes. Last, video number 5 explores the variety of treatment options available and how successful they are in trials and in practice.